Okay, in this first session on reproductive testing, we're going to cover the following topics and concepts. We're going to look at why prenatal testing is routinely performed and look at the sorts of diseases that are diagnosed. We're going to look at different routes of fetal tissue or obtaining fetal tissue and how this can be used for genetic testing. We're going to look at the cellular origin of the fetal tissue that is taken and give an overview of some of the genetic tests that are performed on fetal tissue. But in addition to that, we're going to cover all of these things here. We're going to look at some of the prenatal testing, prenatal imaging, so ultrasound. You should all now be well aware of ultrasound. We're going to cover some non-genetic biochemical tests that can be performed to give some indications of diseases, some of which may be genetic, some of which might not be genetic. And then we'll go on to the routes of sampling of fetal material, chorionic villus biopsy and amniocentesis. Then we'll move on to some of the genetic tests that are carried out for specific genetic disorders. So looking at what we're actually trying to detect in utero, we're looking for a combination of congenital defects, so these are abnormalities that occur in utero, some of which have a genetic basis, such as Down syndrome, which is a chromosome duplication disorder. Others may have a non-genetic basis, such as spina bifida. And the tests that can be done can be either genetic or biochemical, and in some cases purely based on imaging. Then there's another group of diseases under the heading of hereditary diseases. These are non-congenital, so these are due to a defect, a mutation in either the egg or the sperm. So these are classic Mendelian genetic diseases. So with the hereditary ones, we often know that they are there and we can look for them. With the congenital defects, these arise in utero from what was a normal conception, and then something goes wrong along the developmental process. That can be a chromosomal loss or gain or something else. So we call these congenital defects. Okay, so one of the first things that is done is to do some in utero imaging techniques, and the one that most people are familiar with was ultrasound, and we covered this a few weeks ago. Typically scans are offered at 8 to 14 weeks, which is primarily a dating scan, and then at 18 weeks an anomaly scan. The anomaly scan primarily looking for things like Down syndrome, but looking at the dating scan we want to know what's the likely date of conception, and based on the size of the fetus that can be reasonably accurately calculated. It can confirm the number of fetuses, and it can confirm some major structural abnormalities of the skull and the spine. At 11 to 14 weeks, you can also do a combined knuckle translucency scan, which is looking for this region here. And this is a fluid buildup at the back of the head, which is this fluid buildup is much more prominent in cases of Down syndrome. So if you see that this region is much larger, then that may indicate Down syndrome, and it will detect about 70% of cases. It's not 100% diagnostic, but if we see this on a scan, then we'll be suspicious of Down syndrome, and then a genetic test for Down syndrome would be subsequently undertaken if the parents wish that to be done. This can also be followed up with biochemical tests, which I'll come on to in a moment, which may further support this. There's also a specific anomaly scan offered at 18 to 20 weeks, specifically looking for structural abnormalities to the fetus, and this is offered at a time that would allow termination within the legal timescales if abnormalities were found, and it allows timing of subsequent genetic testing to confirm the genetic basis of anything that is observed. So if a fetus is picked up at this point in time with a severe developmental or chromosomal disorder, then the parents would be offered that information and given the opportunity for termination. Also, this can pick up abnormalities with the placenta, so if the placenta lies across the cervix, then that would mean that that would require a caesarean section. So things like this can also be picked up on this scan. So not all tests that are done on the developing fetus are genetic. Some of these are biochemical tests, and one of the most widely performed tests is alpha-fetoprotein. So this is a major protein of the yolk sac. It's the major fetal albumin protein, and it's increased in maternal blood if there is an open neural tube defect. So think about diseases like spina bifida. There's a whole range of neural tube defects, and we would see increased amounts of this in maternal blood, and then that may either lead to a scan being done, or it may be that might be being looked for as a result of an abnormality on a scan to further confirm it. Also, we tend to see lower levels of this protein for unknown reasons in Down syndrome. So if we have a combination of nuchal translucency showing some changes out of normal range, increased maternal or paternal age, which is a risk factor for Down syndrome, and alpha-fetoprotein levels being low, then you can combine all these together to give a likelihood of that fetus having Down syndrome even before a genetic test is performed. But for conclusive results, that requires a genetic test for Down syndrome. So if we want to perform a genetic test on the fetal material, or on the fetus itself, we need to obtain some fetal material. And there are two main routes, the chorionic villus biopsy or amniocentesis. So I'll go through chorionic villus biopsy first. So this is removal of fetal tissue from the chorionic villus. This can be done from weeks 11 to 15. And this can be done also depending on where the placenta is. It can be done either via the transcervical route or the transabdominal route. And here we're using an ultrasound to direct a needle or a biopsy to be able to obtain fetal placental material. And you have to remember that part of the placenta will contain maternal cells, part will contain fetal cells, so we want to make sure we are getting some fetal tissue, otherwise the test would be compromised. So the chorionic villus biopsy, it has a risk of spontaneous miscarriage, so it's something that is only done if there's a genuine or anticipated risk. So if we know that the fetus is at risk of something like Down syndrome due to abnormal biochemical markers, or the nuchal translucency test, or older parents, putting them in the high risk category, then that may warrant a chorionic villus biopsy being done. And this can be done earlier than amniocentesis, meaning that any terminations that are performed are done earlier. If it appears that the fetus is not developing properly, then we can look for unknown chromosome abnormalities. So we can karyotype the entire, the fetus's chromosomal constitution. So we call that karyotyping, and we can see a full chromosome full set of chromosomes of the fetus, which may flag up something. And we can perform molecular genetic tests, usually for known / expected diseases. So this is where, if the parents are known to either carriers of a recessive disease such as cystic fibrosis, or one of them is affected with a dominant disease such as Huntington's disease, if we know the parents have got these genetic mutations, we can look for those in the fetal DNA. So this is where we primarily use a chorionic villus biopsy. There's also the amniocentesis. So this is done later. So this would be typically done if something was identified later on in development. Here again, we're using an ultrasound, we're guiding in a needle to take some amniotic fluid. The amniotic fluid contains keratinocytes which are shed from the fetus, so we can take those cells and we can either culture them to perform a karyotype. So that means, to perform a karyotype, we need to culture the cells and to be able to see the chromosomes. We can also perform a FISH test, which I'll talk about later. Fluorescence in situ hybridization, that looks for specific chromosomes, so we can count the chromosome numbers, or we can extract DNA and perform a polymerase chain reaction test to look for very specific mutations that we know that are present in one or both of the parents. For any genetic test that is done, we need to be absolutely sure that the material that we've obtained is fetal in origin, and there's always a possibility of contamination with maternal DNA. So with a chorionic villus biopsy, we're going for a bit of the placenta that we know is most likely to be fetal, but we have to remember that there are maternal blood vessels running through there and other maternal tissues within the placenta. With the amniocentesis, we are taking keratinocytes shed into the amniotic fluid, but by inserting that needle, there's a possibility of picking up maternal cells on the way. So no test is 100% accurate, but they have a very, very high sensitivity and specificity. Now, when we're looking at the fetal origin of material used in genetic tests, it's important to look back at these slides, which you should be fully aware of the development of the zygote through to the 2-cell, 4 and 8-cell stage, through to the morula and blastocyst stage. The fetus will develop from the inner cell mass, which are these cells in yellow, and then we have these outer cells in dark blue and light blue, and the light blue polar trophectoderm is shown there, and we're going to be taking cells that basically come from these what we call the extra embryonic tissue. So the embryonic tissues are going to come from the inner cell mass. The placenta and the extra embryonic tissues are going to come from these outer cells. So we're going to be wanting to sample structures with a chorionic villus biopsy for these extra embryonic tissues rather than actually taking a biopsy from the fetus itself. So is it shown in a different way? The inner cell mass contains the embryonic stem cells, produces all of the embryo and some extra embryonic tissues, whereas the trophoblast cells here will form other extra embryonic tissues. And this is just shown in more detail from the zygote through to blastocyst. We've got the inner cell mass produces all of the cells of the embryonic tissues and some things like the amniotic ectoderm, but the outer cells of the blastocyst produce the majority of the extra embryonic tissues that we're wanting to sample. And you've probably seen all of these slides before, but you can see the inner cell mass there and the trophoblast there. You can see the developing placenta developing here. In a little bit more detail, you can see the different embryonic germ cell layers developing and all of these extra embryonic tissues that are coming from the trophoblast cells. So that's what we're wanting to sample. And specifically, we're wanting to sample the chorionic villus of the placenta, which is embryonic in genetic origin, whereas there are maternal cells just on the other side. So if you think back to the structure of the placenta, and you should have covered spiral blood vessels and various other things, but there was a boundary between fetal tissue and maternal tissue. And if we go in and take a biopsy from the maternal tissue, then we're not going to get the genetic makeup of the fetus. So that's why this is done under ultrasound guidance, and we're specifically looking for some of this material here. Okay, so what we've covered is how to obtain some genetic material from the fetus by either chorionic villus biopsy or amniocentesis, both of which will provide some genetic material that we can do lots of genetic tests on. What I'm going to cover next are the sorts of genetic tests that we routinely perform. So firstly, we can perform karyotyping, which is looking at the full chromosomal makeup of the fetus. And what we do here is we culture the cells. So we have to have live cells for this. We culture them and prepare them in such a way, which I'll explain in a moment, that we can visualize each individual chromosome, and then we can stain them up to create a banding pattern on each chromosome, and then line them up and just look at each individual band. So this is the job that a clinical cytogeneticist will do. And here we've got a male with 46 chromosomes, an X and a Y chromosome. So any deviation from this pattern would be a chromosome aberration. So this is how it's performed. We take cells and they're cultured in cell culture. So that's taking the cells, the fetal cells, growing them in a liquid medium that contains amino acids and proteins and nutrients, and they will grow in plastic vessels. We call that cell culture. We grow them in the presence of colchicine, which is a mitotic spindle inhibitor, and that stops the cells at metaphase when the chromosomes are condensed. So we must have them growing to be able to perform a karyotype. So we perform this cell cycle arrest, and then we fix the cells in usually ethanol and acetic acid. We then make the cells swell up. We make the cells swell up first, and then we fix them. and then we drop them onto a microscope slide and the cells burst and the chromosomes stick to the glass, and you get something called a metaphase spread, which is all of the chromosomes from a single cell that has burst open as it hits the glass, and then they stick to the glass. We then trypsinize them, which is using an enzyme that eats away at the chromatin, eats away at the histone proteins, and then we can stain them up to identify regions where the trypsin has eaten away at the chromosomes more than other regions, and we get this staining pattern, this black and white staining pattern. It's really actually blue and white, but we always look at it in black and white. And this gives a classic banding pattern for every single chromosome. So that's a chromosome 1, so is that one, there's a chromosome 7, there's an 11, there's a 5, and there's a 3, and a 17. So a clinical cytogeneticist will be able to just look at this image, identify every single individual chromosome, and then using a little bit of Photoshop type software, line them up side by side and check every single band to make sure that every single band is present, and also count the chromosomes. And what we can see here is a case of a male Down syndrome, because we've got an additional chromosome 21. So Down syndrome's the most common, one of the most common chromosome aberrations. About 1 in 100 live births, much more common with older parents. So this karyotyping is sort of an older technique that can be used to confirm the biochemical or test results. So biochemical test or scan results, sorry. Now, we wouldn't necessarily do a karyotype for Down syndrome now. We'll do a FISH test. I'll come into that in a moment. But if there's a chromosome aberration, if there's a suspected chromosome aberration with a fetus that's not developing properly, we may well still do a karyotype like this, because that would flag up any chromosome abnormality on any chromosome. Whereas with the FISH test that I'm going to talk about in a moment, that just focuses on counting the number of individual chromosomes that we are specifically looking for. Usually chromosome 21, X, Y, 13 and 18, and we tend to ignore all the others. So a karyotype gives you the chromosome number and structure of every single chromosome, whereas the FISH test will just investigate specific chromosomes that are frequently involved in developmental disorders, but that will make sense in a moment. Now, there are a whole range of other common numerical chromosome abnormalities. So Turner syndrome, which is loss of an X chromosome, features involving infertility and short stature, particularly in Turner syndrome. Klinefelter syndrome, so 47XXY, often reduced infertility, altered sexual characteristics. We've got triple X syndrome, borderline whether there's pathological features with this particular one. The more severe ones are Tau syndrome, with many physical and mental abnormalities, and Edwards syndrome. So trisomy 13 is Tau syndrome, Edwards syndrome, trisomy 18, which is lethal early in life. So Down syndrome, Tau and Edwards are the three main severe diseases that are looked for. Now, most of these are looked for postnatally, mostly because you wouldn't necessarily see anything on a scan to suspect any of these. So these are things that are commonly observed, not that much rarer than Down syndrome, but they tend to be postnatally analysed because we've got no reason to suspect them. Okay, so I'll now go through fluorescence in situ hybridization, which is a technique that we can use to identify a specific chromosome within a cell. So fluorescence in situ hybridization, it basically involves creating a chromosome preparation on a microscope slide. We denature that DNA, so we dip the slide into a denaturing solution such as formamide at about 70 degrees, and that will cause all of that DNA that's stuck to the glass slide to denature to single-stranded DNA whilst remaining its general shape, chromosomal shape. And then we apply a DNA probe. There's a few bits on here that maybe are not too relevant, but we get let's keep it simple, a single DNA probe. And that single DNA probe could be just the centromere of a particular chromosome. So, for example, centromere of chromosome 21, and we could take DNA that represents the chromosome, the centromere of chromosome 21, label that red, and then apply it to the denatured DNA that's on the microscope slide. And if this red labeled DNA probe binds to the centromere of chromosome 21, it will stick, and that will give a red signal. And we can do that for any region of DNA. So we can just buy these probes off the shelf. They are purified bits of individual parts of chromosomes labeled either red, green, blue, yellow, various different fluorescent colours, and we can buy them, apply them to our chromosomes on slides, see where they stick, and then use that to count chromosomes. So the workflow is fix cells to the microscope slide. These can be from amniocentesis. Denature the DNA, apply the fluorescently labelled DNA that's homologous, as in the same sequence as the desired chromosomes, wash off the excess, visualise under a fluorescent microscope, and then we counterstain all of the chromatin blue with a dye called DAPI. That binds to total DNA, and what we're looking for is blue chromatin with red, green, or other coloured spots on there to identify the location of specific chromosomes. So in this particular example, we don't actually need growing cells for this. Whereas we did for karyotyping, we needed cells which are alive and cultured and stopped at metaphase. What we can do here is take cells in interphase. So that's the part of the cell cycle that cells are in most, G1S and G2 of the cell cycle. And in interphase, the nucleus just looks like a fuzzy blue blob because all of the chromosomes are decondensed and all mingled up together. What we do is with that cell that's stuck to a microscope slide, the DNA is being denatured. We've applied our probes, in this case a red probe that recognises the centromere of chromosome 21 and a green probe that recognises the X chromosome, and what we can see is that this is a female Down syndrome case. What we can also do is combine this with multicolour fluorescence in situ hybridization, so use multiple probe sets simultaneously, and the chromosomes themselves don't show very well because they're not stained with DAPI here, but what you can see is we've stained this cell, and it's actually a chromosome spread with 5 or 6 different probe sets. So in blue we've got 2 markers there, which is chromosome 13, so that excludes Fetal syndrome. Purple excludes Edwards syndrome because there's 2 spots there. Yellow is chromosome 21, there are 2 spots, so it's not Down syndrome. We've got an X chromosome in red and we've got a Y chromosome in green, so this is a normal case. So this is a sort of fish test that can be done to rule out all of the common chromosomal numerical abnormalities. And if you check through, I think this one is showing exactly the same thing, only in interphase fish. Now, we've not used the DAPI stain here because it interferes with the blue, but that is an interphase, that's a metaphase. And if you alter the contrast of the image, you can just about make out the other chromosomes here that have not been stained. Okay, so everything I've been talking about so far is looking for chromosome abnormalities, and chromosome abnormalities are typically congenital abnormalities. They occur in utero from what was a normal conception, or they can occur due to an abnormal egg or sperm with an additional or loss of a chromosome. But they are large genetic abnormalities. We're now going to move on to molecular genetic tests, and molecular genetic tests are looking for mutations often at the single base pair level. So the order of magnitude here is much, much smaller. We're dealing with single base pair changes or just small changes within an individual gene. So here we're dealing with all what we call the classic Mendelian genetic diseases, the ones that are caused by inherited mutations, so your cystic fibrosis, Duchenne muscular dystrophy, Huntington's disease, all of the major inherited diseases. Now, without knowing the causative mutation in the parents, you can't do the test. So there would never be a situation where parents would have a chorionic villus biopsy with no family history of cystic fibrosis and request that be tested for cystic fibrosis. There is no point doing that. If the parents are at risk, they would be tested. We then need to find out exactly what mutation the parents have got and then look for those same mutations in the fetus. Now, unlike chromosome analysis, we just need a very, very small amount of DNA. We certainly don't need growing cells. We can do this on a handful of cells. Now, in topics in human biology, I've talked about polymerase chain reaction briefly at the start of the module. Polymerase chain reaction is a method of specifically amplifying a very small region of the genome, and we do this using a combination of DNA polymerase and DNA primers. So, if we imagine this is the region of DNA that we are interested in that might harbour a mutation, we can specifically amplify that DNA and create millions of copies of it so that we can then analyse it on a agarose or polyacrylamide gel and then subsequently look for the presence of the mutation. So the first thing that we do with the cells is extract the DNA and perform polymerase chain reaction. We design some short oligonucleotides. So these runs of about 20 nucleotides which match the sequence here and match the sequence here. And the combination of DNA polymerase will extend this primer in this direction, and DNA polymerase will extend this primer in this direction to create copies of that piece of DNA. What we then do is heat denature the DNA, allow it to reanneal, and allow more primers to bind and create. So this primer will bind here, this primer will bind to the newly produced strand from there, this primer will bind to a newly produced strand from there, and this primer will bind to the original template. And at the end of the second cycle, we've doubled up our DNA again. So we've doubled our DNA for this specific region in the first cycle, and in the second cycle we double it again, and then in the third cycle we've doubled it once more. And this doubling of DNA just for this region of DNA between this forward primer and this reverse primer, this region gets selectively amplified so that we have millions of copies of that DNA. Once we've got millions of copies of that DNA, we can then analyse it on some sort of gel and look for the presence or absence of a mutation. And that mutation that we're looking for is the mutation that is present in one or both of the parents. So if you don't fully understand PCR, look back on when I talked about PCR in topics in human biology, and there was a PCR tutorial that I went through. So with the polymerase chain reaction, we are using DNA polymerase, we're using DNA primers which bind to regions close to the mutation. We then heat the DNA and cool it between 95 degrees to denature the DNA, 60 degrees C to allow the primers to bind, and 72 degrees C to allow the thermostable DNA polymerase to extend from the primers. We repeat this cycle for 30 times, so 30, 95, 60 and 72, repeat that 30 times, and we end up with millions of copies of that DNA which can be used for any sort of mutation detection strategy, which I'll not go into the details of here. But this is the basics of what we call molecular genetic testing, different to cytogenetic testing because we are looking at the individual base pair level of the DNA as opposed to huge chromosomes made up of hundreds of millions of base pairs. Okay, to summarise everything that we've covered, prenatal diagnosis involves a combination of imaging, biochemical tests and genetic tests. If we're going to perform a genetic test, we need DNA from fetal tissue, and the main routes are chorionic villus biopsy and amniocentesis, and both of these have a risk of miscarriage, so we only do these if we suspect that there is a significant risk to the fetus of having some sort of genetic abnormality. Sometimes suspected defects are not identified until late in gestation, maybe after an anomaly scan, so that might lead to a fairly late amniocentesis being performed, but the amniocentesis is performed at a time that then allows termination to be performed before that maximum date for termination of 22 to 24 weeks. So some congenital and some hereditary diseases can be diagnosed using prenatal testing. In the next two sessions, we're going to cover more extensively prenatal testing and particularly focusing on the ethics of prenatal testing, looking at what diseases are okay to test for with the aim of performing a termination and what genetic changes are not okay to test for. We've also got non-genetic non-genetic diseases that can be picked up, such as neural tube defects, and they tend to be picked up by imaging and or biochemical tests. And it's also worth having a look at these two links here. These are two links talking about fetal DNA testing in maternal blood. This is something that I will cover in the fourth session. So So this is the first session. The second session will be prenatal genetic testing and ethics. Session 3 will be pre-implantation genetic testing and ethics after IVF. And then session 4 will have some reproductive advances, and one of the reproductive advances will be fetal DNA testing in maternal blood, where DNA from the fetus crosses the placental barrier, ends up in the maternal blood, and we can detect that using some advanced genetic testing. So have a look at these articles here, and I'll cover the theory of that in session 4. Okay, that's the end of this session.